Let us begin with asexual reproduction in unicellular organisms. Unicellular means those organisms which are made out of just one cell. All the life processes are completed by just that one cell organism. So the first method is called binary fission. Binary means two, and fission, that is breaking or splitting into two or more parts. So this method is shown by prokaryotes, that is bacterial cells undergo this type of division. Protists or protozoans like amoeba, paramecium and euglena show this type of method of undergoing splitting and forming new cells. And eukaryotic cell organelles like mitochondria and chloroplast perform asexual reproduction by binary fission. So even cell organelles like mitochondria and chloroplast undergo binary fission. That is, they divide by this particular method. In this process, parent cell divides to form two daughter cells, and both of the daughter cells are going to be similar to one another. Binary fission occurs either by mitosis or amitosis. So binary fission can occur by both of these methods. Mitosis we have already studied in detail, which we know that occurs by karyokinesis and cytokinesis as two main stages. In karyokinesis, there is nuclear division, and which takes place in 4 steps. We had seen about prophase, metaphase, anaphase, and telophase. Then comes cytoplasmic division through cytokinesis. So karyokinesis, that is nuclear division, and cytokinesis, that is division of the cytoplasm. It's a very systematic method that is involved in mitosis. But amitosis is random distribution of genetic material between daughter cells. There is no systematic division of the parent cell into daughter cells. So cell division by simple cleavage of the nucleus followed by division of cytoplasm without spindle formation or appearance of chromosomes. So it's a very generalized or random distribution. The nucleus is going to get divided into two halves followed by the division of the cytoplasm without any spindle formation or the appearance of chromosomes. So this type of amitosis is generally found in prokaryotes, and some protists like paramecium will show this type of method of division, that is amitosis. Let's see the very first type of binary fission. There are 3 types of binary fission. The very first is called simple binary fission, subdivision of cell. Fission, that is subdivision of cell in amoeba. The example is amoeba. Axis of fission or division is through any plane in amoeba. Now we know that the body of amoeba is irregularly shaped. There is no fixed shape of the body of amoeba. Hence, the axis of fission, that is the line through which the organism's body can be divided into two halves, is not fixed. Hence, the axis of fission or division is through any plane in amoeba. Binary fission is performed during favorable conditions like abundant food. Generally, we find the amoeba in stagnant pools and water. So when the water and food material is available, amoeba undergoes division by this method, that is simple binary fission, where the body of amoeba can get divided into two halves through any plane. There is no fixed plane, horizontally, vertically, diagonally. There is no fixed plane through which it can get divided. So through any plane the body can get divided. So here we can see a single parent cell with a nucleus and cytoplasm. It has started dividing. The nuclear division has already started taking place. Two nuclei, you can see cleavage furrow has already started forming, which keeps on deepening, and this cytokinesis is also complete. Cytoplasm has also divided. Nucleus has also divided and migrated towards the two cells, and finally you can see two new daughter cells that are formed. So this is one type of binary fission, that is called simple binary fission, which we observe in amoeba. Let's see 2 more types of binary fission. Here we have transverse binary fission in paramecium, which is another protist. But here, unlike amoeba, the plane of division of the parent body is fixed. So it is called transverse binary fission. The body gets divided into two transversely, and hence the name transverse binary fission. So here we have the diagram for paramecium, and the nuclear material gets divided. Cleavage furrow has started forming, and transversely you find that the body is getting divided into two halves, and you have two daughter paramecium over here. So unlike amoeba, where there is no particular or fixed plane through which the body is going to get divided, but here we find that the body of paramecium gets divided transversely into two halves, and we get two daughter paramecium. Let's see the third method of binary fission. The third method is longitudinal binary fission, which is found in euglena. So this is the diagram for euglena. It's green in color, so it has got chlorophyll, so it can synthesize its own food, but it is not a plant because it has got a flagellum, which is a hair-like structure, and it is used as a locomotory organ. Since the organism is capable of locomotion, it is included in the category of animals. So paramecium shows longitudinal binary fission. That is, the body of the organism is getting divided into two longitudinally or vertically into two halves. So these are the various stages in which we find that the animal gets divided. So gradually from top, the cleavage furrow gets deepened, and flagellum also divides. You can see two flagella now, and nuclear division also taking place. And finally this furrow cleavage becomes deeper and deeper, and it gets divided into two, giving rise to two daughter cells. So these are in short the three methods of binary fission, that is simple binary fission in amoeba, transverse binary fission in paramecium, and longitudinal binary fission in case of euglena.